Click for IB Biology. Here we're looking at cell division as an overview initially before moving on to the stages of mitosis associated with cell division in eukaryotic cells. There's a blank PDF of this presentation available in the description, which you can annotate with your own notes. There's a quick quiz, there will be some channel links, and there's an AI prompt which will allow you to do your own self-assessment. So let's take a look at the phenomena of cytokinesis, which means essentially cell splitting. And our overview is that we have the original cell, which is often referred to as the mother cell, and this will undergo mitosis. Our discussion is largely about mitosis, although we do carry the idea of prokaryotic binary fission as well. So we have mitosis, and then the stages of mitosis occur, and the parental cell, the mother cell, will divide into two so-called daughter cells. And of course, in my image, I've tried to indicate that the two daughter cells, of course, will certainly initially be smaller than the mother cell before going through a growth phase. So the splitting process is called cytokinesis, and is not actually part of the mitosis process. It follows mitosis. And it follows it in two different styles, depending upon whether you're an animal cell or a plant cell. Let's look at the former. The former shows that the two cells, as I'm trying to show in my diagram here, form what is known as a cleavage furrow. A contractile ring of proteins forms around the equator of the two cells, diagrammatically shown here, and that contractile ring is formed from actin and myosin proteins, along with what are called motor proteins. And so what happens is that with the contraction of this ring, the waist between the two cells becomes narrower and narrower until there's self-sealing of the cell membrane and the division of the cell into the two daughter cells, which of course will be smaller than the original parental mother cell. Contrast that with the cell division of a plant cell, which is necessarily different because of the presence of the cell wall. And we have this so-called phragmoplast vesicle structure, where vesicles begin to form across the equator. Rather than the actin-myosin cleavage furrow, we have these vesicles that form across the equator. So diagrammatically, I'm showing that here. Now, just to add an important point, both the animal and plant version, cytokinesis, follows what is called the telophase, the last phase of mitosis. So this is after the telophase. And so we have the two nuclei here, and the vesicles form along the equator, and then the vesicles will merge together until there's an entire division across the center of the plant cell, upon which the plant cell will secrete outside of its cell membrane cellulose fibers for the construction of the cellulose cell wall. And in this way, the division of an animal and a plant cell is quite different. The diagrams above that we just looked at imply that the division shows the cells producing two equally sized daughter cells, but this is often not the case. And in the case of the formation of the oocyte, which is the mammalian egg cell, cell division is very much unequal cytokinesis. And so the first cell to go through the division in this particular case forms one large cell and one small cell, which is not entirely separate and is in fact known as the polar body. And this polar body has no cytoplasm, no organelles. Those are all retained within the larger cell here of the oocyte. And in fact, we have what's called the first polar body, and the main cell will actually go through another division to form a second polar body, again with unequal cell division. Now, you may have already studied the oocyte as a specialized cell, and you'll recall that what's going on here is that largely this process is trying to create a very large cell which has a large volume, and in that volume it contains the organelles for the new organism once fertilized, and also the nutrient necessary to support the development following fertilization. And so the organelles, of course, would include such things as the mitochondria. And so that's the reason for the uneven division, is to have a larger cytoplasmic volume with all the organelles confined to one cell. And here's an actual image of the structure at 140 micrometers. You perhaps remember the sperm cell has a diameter of the head structure of 5 micrometers, although the tail is much, much longer. Another example of unequal division is found in the fungi called yeast. Yeast, of course, we will know from the brewing industry and from the bread making industry. Yeasts also are associated with a number of infectious diseases, and they also form unequal cell division, which we can see diagrammatically here as we go through the cytoplasmic division, in which the bud is initially much, much smaller than the original. So there's not an equal division, but an unequal division to form the smaller, one of the cells is the smaller daughter cells, and then those in turn will go through other divisions, making smaller daughter cells, which ultimately will increase their size. So that's 2 examples of unequal cell division. Another overview fact of mitosis and meiosis to consider is that mitosis maintains the chromosome number. So the number of chromosomes in the nucleus of the mother cell or parental cell is the same as the number of chromosomes in the 2 daughter cells. And of course, you'll be aware perhaps that this is a consequence of a process called DNA replication. Now, this is true of mitosis. Now, interestingly, we often think of 2N providing 2 daughter cells also with the same number of chromosomes. So let's take a human example. That would be 46 chromosomes here, meaning that we have to have 46 here and 46 there. And that's the relevance of the semi-conservative DNA replication process, which I think is covered in D2.1. And so we maintain the chromosome number, which is the 2N to 2N. However, the interesting thing is that there is an example where mitosis will also conserve the haploid number N to N, and this is found in the gametophyte stages of reproduction in organisms such as ferns and mosses. So there's a slightly different life cycle for those organisms in which mitosis plays the process of maintaining the chromosome number, but we go from haploid to haploid. We're more familiar with the diploid to diploid. Now let's contrast that with meiosis. Now meiosis, we begin with the mother cell. Now the mother cell, of course, in meiosis would be found in the reproductive organs of the male, or indeed of the female. So in the male, this would be in the testes, and then the female in the ovary. And so this parental diploid cell, what happens is that the number of chromosomes in the process of cell division is halved. And so the number becomes halved. And so we reduce from the diploid number to the haploid number like this. Now there are a number of details to this meiosis process which are not revealed by this simple diagram. One of which I will introduce now is that essentially one should think of meiosis as reducing a double set of chromosomes, a double set of genes, to a single set of genes. So in a human being, the process of meiosis reduces the double set of our diploid cells with a double set of its chromosomes to a single set of its chromosomes, a single set of its genes. So we'll return to this in more detail and clarification in the process of meiosis. The rest of the videos in this series are associated with mitosis, and the details of that process. Here are the links here. Subscribe to the channel, please. And there's a link to the whole playlist.